Hey guys, welcome back to another video made by COGO. Today we're going to talk about PCR, which stands for polymerase chain reaction. Today we're going to talk about how PCR works, what the applications of PCR are, and how PCR can be applied in COVID testing. PCR is a method in biology where it aims to amplify, meaning making a lot of copies, of a particular segment of DNA. This method has many applications to it in the real-world situation. Now, exactly how does PCR work? Well, first of all, there are 3 stages to PCR. The first stage is denaturation, which happens at around 96 degrees Celsius. The second stage is primer annealing, which happens around 55 degrees Celsius. Now, the last and final step is primer extension, and this happens around 72 degrees Celsius. This is done within a mineralized saline solution, and the original DNA sample is added into the solution. Now, Taq polymerase, primer, and nucleotides are also added into the solution. First of all, we have denaturation, which happens at 96 degrees Celsius. In this stage, DNA is heated up to extreme temperatures and strands are separated. Now, the second stage, called annelation, happens at around 55 degrees Celsius. After the DNA sample has been brought up to extreme temperatures and separated, this mineralized saline solution with the DNA sample is cooled down. Now, it is important to note that primer is purchased in bulk and added in surplus into the DNA samples in the mineralized saline solution, which is just simply salt water with some stuff floating around. The primers bind to complementary ends of DNA regions that are being copied, and the sequence of the primer is selected during the purchase. The third and final stage of PCR is primer extension, and this stage happens at around 72 degrees Celsius. The solution is heated up again, and Taq polymerase, which is very heat resistant, is added into the solution. The role of the Taq polymerase is to extend the primer segment by adding nucleotides to that strand. After going through the 3 stages of PCR, this DNA segment is successfully replicated. The number of DNA molecules will double after going through 1 cycle of PCR. An individual is able to perform the 3 stages of PCR many times, but they typically end on the 35th time in a lab environment. Therefore, technically the number of DNA molecules after 35 cycles would be 2 to the 35th power of molecules. There are numerous applications of PCR within the real world, but some of the most practical applications include cloning DNA into a plasmid for further experimentation, or forensics, which basically matches small amounts of DNA in a crime scene to DNA in databases. There are also medical diagnoses, which check for genes of predisposed medical conditions or hereditary diseases. These also check for diseases such as COVID. Now, the last application in PCR is gel electrophoresis. This separates DNA, RNA, and protein molecules based on size and electrical charge. This is extremely useful within a lab environment. RT-PCR, which stands for reverse transcription polymerase chain reaction, is often used in COVID testing. This type of PCR uses a special polymerase called RT transcriptase, and this can convert the protein into DNA, which can later be used in PCR for amplification. PCR tests, including RT-PCR, are the fastest and most reliable methods of pathogen or virus detection. Thank you so much for watching, and I really hope that you enjoyed this video. Always remember to never stop learning, and I'll see y'all in the next one.